Good morning, everyone. Welcome to today's webinar on DNA barcoding. I am Dr. C.P. Prakash, heading the Department of Plant Biology and Biotechnology from Sri Chandraprabhu Jain College, Minj. I am so much happy to have you join us today. Thank you for taking the time out of your busy schedules to explore this topic. In next few minutes, we will diving into the topic. This webinar aims to provide you with inside knowledge about DNA barcoding and how technology developed and how the biology field overcome all other departments. In this time, I thank our management members, principal, heads, and staffs of our college, and District Chief Guest today's Chief Guest Dr. Prasanna Sir, to joining with us. During the session, feel free to ask questions. You may have comment your questions in the chat box. If you can't make the live session, don't worry. A recording will be available in our college YouTube channel after the event. Without further delay, let's begin the program.
All right, sir. Can I start, sir?
Wait, sir.
Yes, sir.
We will go for presidential address now. I request
Yes.
our principal, Dr. N. Sujata ma'am, to presidential address.
Very good morning and warm welcome once again on behalf of Sri Chandraprabhu Jain College. I am Dr. Sujata, heading the institution. I am very glad and congratulate the Department of Plant Biology and Biotechnology Department having organized the webinar in this academic year 25-26 with a kickstart. I glad and happy welcoming Dr. Prasanna Kumar for your presence and accepting our invitation, sir. So this is a topic. It's a new topic for us also, DNA barcoding. I hope the audience will get a enriched knowledge after this webinar by your inputs given. So I too new to this topic, so we very well eagerly waiting for your session. Thank you all once again for joining in this session. I thank all the participants for joining for this webinar. I thank the management and all the faculty members and the students, those who are present today for this webinar. Thank you, Anna.
Good morning, everyone. This is Dr. Bharat. I'm going to propose a speaker introduction. We are delighted to welcome Dr. Prasanna Kumar C as the speaker for today's session on DNA barcoding. Dr. Prasanna holds a PhD in marine microbiology from the Center of Advanced Studies in Marine Biology, Annamala University, supported by the prestigious DST Inspire Fellowship in the year of 2013. He later pursued postdoctoral research at Xiamen University, China in 2017, funded by the National Postdoctoral Foundation of China. He has served as a research associate at CSIR National Institute of Oceanography in 2020 and was entrusted with maintaining India's marine biodiversity database during his tenure. His journey in DNA barcoding began in 2009 when he joined the Census of Marine Life project funded by the government of the United States as a junior research fellow. The project involved synthesizing DNA barcodes for a wide range of marine flora and fauna. Since then, Dr. Prasanna has successfully barcoded over 300 marine and terrestrial species. In addition to his research contributions, he is the co-founder of Barcode Biotechnologies, an emerging bioinformatics and sequencing initiative currently operating in Goa, Hyderabad, Bangalore, and Chennai. Dr. Prasanna has also played a key role in establishing two recognized microbial culture collection centers at National College, Trichy, and Kristu Jayanti College, Bengaluru. He is currently serving as an associate professor in the Department of Life Sciences at Kristu Jayanti College, Bengaluru, when he continues to inspire students and researchers in the field of molecular biology and biosecurity. Let's extend a warm welcome to our Dr. Prasanna Kumar and look forward to an insightful session. Thank you. So you can start, sir.
Yes, sir. Thank you. Thank you for your warm introduction. Respected Sri Lalit Kumar O. Jain, Secretary, Sri Chandrababu Jain College, respected Dr. N. Sujata, Principal, and respected Dr. C. Prakash, Head of the Department of Plant Biology and Plant Biotechnology. Thank you very much for this opportunity to, it's my favorite topic, DNA barcoding. So thank you for this opportunity to share my views on DNA barcoding. So I understand most are, I think, from botany background, listening from undergraduate and postgraduate level. So I will just start with simple terminology. What is this barcoding? Okay. Um, See, you have a universal product code. Every product, once it is synthesized in the factory, their details, date of manufacturing, everything has been converted into binary barcodes, and it be stored in the database, and the corresponding barcode will be pasted on the product, so that when they reach a supermarket, just by scanning the UPC, you will come to know what is the information about the product. Similarly, every living system, biological species, have a DNA sequence which can be used to identify their species. Okay, so the DNA barcodes work parallel to universal product codes. By recognizing and reading these DNA sequences, you can identify the species. That's the species identification is an entire science behind DNA barcoding. Okay, so I want to insist why DNA barcoding is required. Okay, here is I have made a big cursor so that I can point out to the graphs. Here is a graph. You can see it is broken here. We have nearly 8 million species in this biosphere. 8 million different species. Homo sapien is one among those 8 million species. Among these 8 million species, with the last 200 years of scientific research, we have able to document only 1.5 million. Okay, not more than that. Meaning, thousands of species in this biosphere are evolving and going extinct without being documented. Right, we are losing biodiversity in alarming rate, and most of the biodiversity we don't even realize that we are losing because we never document them. That's the facts happening. So it's very clearly we need a rapid technology to document species, to document biodiversity around us, and also in rare ecosystems. So that's why DNA barcoding. Okay, DNA barcoding, is it very new concept? No. In microbiology, people know that we've been using DNA sequences for more than 45 years now to identify the microbial species. But for zoology and in botanical science, we've been using DNA sequences, but not a common one for all plant species. Okay, for example, angiosperms, they are widely using ITS, internal transcribed spacer region, for identifying the angiosperms. And for gymnosperms, other set of sequences were used, and never ever you can compare the DNA sequences of angiosperms or gymnosperms. Okay, so in order to make it into a narrow channel, in order to uniformize this effort of sequencing and identification, uh, yeah, scientists came up with this idea. The entire idea works on this principle. Okay, first you identify a species. Yes, you need a conventional taxonomist to do that. First, identify the species, sequence a particular gene. Here we call them as a DNA barcode. Sequence that, deposit in the database, so that when a non-taxonomist, when he doesn't know how to identify the species, can take the species, sequence it, compare it with the database, he will come to know what species it is. Okay, so initially the effort was to identify one particular gene that can be used for identifying both animal, plants, and microbes, but turns out it is impossible to identify one gene that is being present in all domains of life. So these are the four genes now are currently used now. Okay, for animal identification, the mitochondrial gene, cytochrome C oxidase 1, CO1 gene it is called, it is being used as a DNA barcode. For plant species, we are using RBCL gene and MATK gene, Maturase K and ribulose 1,5 bisphosphate carboxylase large subunit. This is RBCL gene. Okay. Mostly for angiosperms, Maturus K is used, and gymnosperms RBCL is preferred, but it's not a hardcore rule. For some plant species, you have to sequence both the MATK and RBCL gene for its identification. For fungi, they don't have even COH genes in mitochondrial region of the fungi is not sufficient to act as a barcode. So for fungi, the recognized barcode now is internal transcribed spacer region. This ITS gene is being used for fungi, and for last 2 decades, I have shown that I have told that the 6NS RNA has been widely used for prokaryotic identification. So what these 5 sets of genes are doing is, using these 5 sets of gene, the entire biodiversity in this biosphere can be decoded. You don't need to be a taxonomist. Okay, so this is the facility DNA barcoding, and it is very rapid too. Maybe you guys are UG students, you may be very young to realize this. 20 years back in India, if you want to identify a plant species, you need to approach a Botany Authority of India. Okay, they have to authenticate your sample as belonging to particular species. Without their authentication, you cannot go forward any scientific communications. Okay, which has been completely changed now. Even non-taxonomists can identify the plant specimen to species level using DNA barcoding. Or in other way of saying, these days all scientific communications are demanding DNA data when you are claiming a biological specimen to be as a particular species. Okay, so that's what DNA barcoding has done. How this barcoding works, this is a simple diagram. Okay, so once you are working with a bamboo leaf, you are identifying the bamboo species, extract the DNA, sequence RBCL or MATK gene from the bamboo leaves, store the sequences in the database. Okay, now say bamboo species A have this sequence stored in the database. Someone somewhere in Australia working on the same bamboo species A, if he want to identify, he is not a taxonomist, but still he can identify because simply he can produce the sequence now. We have online softwares like BLAST tools offered by NCBI. When you put your sequences there, the sequences will be matched with the database. Wherever it is getting 100% match, that species name will be displayed for the non-taxonomist. That's how the entire barcoding works. You build the database slowly. Once the database is built, you can use the other sequences, the sequences of unknown species to compare with the database, and you will find the species name. So the entire idea of barcoding is proposed by Professor Paul D. Herbert. He's still active in service. He belongs to University of Gulf. He just not stopped after proposing. He published multiple papers, and he is the responsible guy for initiating multiple consortia. The first consortia he initiated was International Barcode of Life. From there, they went on Bulgarian barcodes, even Indian Barcode of Life. We have a network of barcoding people, Zoological Survey of India, Botanical Survey of India, and many, many more institutions are playing an active role in decoding our own biodiversity. We have UK Barcode of Life, San Diego, German Barcode of Life, Norway Barcode of Life, Australian Barcode of Life. So these are the various consortia which took the responsibility to decode their own biodiversity in their vicinity. It's not only geography specific, they are also domain specific. For example, we have Fish Barcode of Life, which dedicatedly focus on both freshwater and marine water fishes. We have Census of Marine Life, where I have worked as this is my first project. Under them, I was responsible to decode the Indian Ocean biodiversity, where they have launched a barcoding marine life consortia, which focuses on decoding the marine biodiversity around the world. Okay, Indian Ocean chapter, we took care of it. And we have barcoding US ants, we have barcoding wildlife, Lepidoptera barcoding, sponge barcoding, all birds barcoding initiative. Likewise, we have various barcoding initiative because, like I said, once you built a database, then only the barcoding will be very much effective. You need to have a database with a wide range of species barcodes in them. Otherwise, for non-taxonomists, it will be very difficult to identify the species. So the entire barcoding initiative have started during 2003 with the publication of this paper, Biological Identification through DNA Barcoding. And by 2009, 6 years after, they proposed RBCL and MATK gene as a plant barcodes in this paper. If you read the paper here, you will see they have used more than 53 genes, potential genes in the genome of a plant, and they came with a conclusion after analysis that for angiosperms, Maturus K is working fine, and for gymnosperms, RBCL is working fine. But for many cases, you need to sequence both RBCL and MATK for precise plant species identification. Okay, and subsequent year they came up with a paper of proposing that CO1 may not work fine for fungi, so they have proposed an alternative barcode, internal transcribed spacer, that can be used to identify both yeast and molds of fungal group. And by 2013, we revalidated this paper by testing this effect of ITS gene sequences in marine fungi. We have produced around 1,200 barcodes and belonging to 77 different species of fungi from marine drilling. So this is a national scenario in India. I would say this Department of Marine Biology, Center of Advanced Studies in Marine Biology in Annamalai University, they got this project from consortium of Barcode of Life in 2007. From there, the barcoding effort was amplified in India. The first barcoding paper from India is around 2009, published from Annamalai University. This is on marine finfish barcoding. It is in current science. You can access those papers. So the subsequently Botanical Survey of India took charge of plant biodiversity, and Zoological Survey took charge of terrestrial animal diversity. And National Bureau of Fish Genetic Resources and CMLRE, Central Marine Fishery Research Institute, took an initiative on barcoding marine flora and fauna thereafter. And Center for Marine Living Resources in Kerala, they are now in charge for deep sea barcoding. The organisms are they are exploring the deep sea ecosystems and they are decoding the diversity. So this is a consolidation of national and regional effort and taxon specific working groups that are working on various groups of barcoding. Okay, so what they are doing with the DNA sequence that is being generated worldwide. Okay, in worldwide, the DNA sequence data generated are deposited here, Barcode of Life data system. This database, unlike NCBI and DDBJ, EMBL, these are regular databases that collect DNA sequences, but Barcode of Life data systems, they collect only DNA barcodes, meaning those 4 sets of genes, ITS, RBCL, MATK, CO1 gene, they are being collected here and deposited here so that any non-taxonomist who want to identify, say for example, you are walking through a forest, you are having a leaf, you have no idea about the taxonomy, but you want to identify the species, it is very well can be done very easily. Send it for sequencing, get the RBCL MATK data, put the data here, they will compare the data with your database. In 30 seconds time, you will know the species name. And another proud thing I would say after maybe Operation Sindhu is this Indian Biological Data Center, where in India we have an upcoming database which is storing national nucleotides, the nucleotide sequences that have been generated in India. Indian biodiversity data and other gene sequencing data are now can be deposited in National Nucleotide Data Archive in India, and you can get an accession number, which is playing a similar role where like NCBI and DDBJ. So how does this barcoding works? I told you we need to create a local library. We need a traditional taxonomist, conventional taxonomist to identify the species, document the morphological data, then go for DNA extraction, sequencing, and deposit the sequence with the species name in the database so that when a non-taxonomist access it, they can find it. How they are finding it? Say, if this is the DNA sequence I have deposited in the database, this is a DNA sequence I generated, I don't know what species it is. If this sequence has to be identified as the species for this sequence, they will be matched nucleotide wise. Okay, they will be aligned or matched. The query sequence, the sequence you generated for the unknown species is a query sequence. Reference sequence is a sequence that is there in the database. So the query sequence, each and every nucleotide will be matched with the reference sequence, and when the match is 100% or 99%, the same species name can be displayed. Okay, so the flow initially I told is very simple, but actually it's a very tedious process, and it requires multi-dimensional skill set in barcoding. I will tell you how. Say for example, if I'm working with a plant species, when I want to produce a barcode for the plant species, I need to document meticulously the morphological features of the plant species. So you need the skill of conventional taxonomist, document it, multiple photographing, okay, and along with the metadata, and store it. Then you subject the same specimen for DNA isolation, extract the DNA, go for PCR. It is only in the PCR, in DNA extraction, the entire genome of an biological system will be isolated. It is only in the PCR using a specific primer we are going to amplify the RBCL or the MATK gene. So after amplifying, we will go for sequencing. The sequences are ready now, along with the metadata in case of BOLD, Barcode of Life database, along with the metadata, the morphological data, whatever we have collected, the sequencing machine output file, everything has to be submitted. So after getting the sequences of the barcode region, they will produce something like this. This is just an anonymous barcode. They looks like a UPC product code where they have 4 colors instead of black and white. All 4 colors indicates the 4 nucleotide: adenine, guanine, thymine, cytosine, ATGC. Okay, so they will just produce a synonymous barcode. They have now no role in species identification. The species identification happens by when you search the BOLD database with your query sequences. The query sequences will get matched with the reference sequences, and they will tell you what species it is. So I told you it is a multi-skill set, multi-dimensional skill set is required. Starting with a conventional taxonomist, you need to be a molecular biologist to successfully handle DNA and do the PCR. You need to be a bioinformatician. Once your sequencer gives you the machine output file, you should know how to analyze the machine output file and convert into a refined data. You need to be a data analyst to maintain the database, submit the sequences, and many more. Okay. So, like I said, during DNA isolation, entire genome is isolated. When I say genome, it is a complete genetic makeup of an biological system. Say, for example, Orisa sativa, when I go for extracting DNA from the rice leaf, I will extract the entire genome. They may have more than 50,000 genes. Can every gene act as a barcode? No. The answer is no. For any gene to act as a taxonomic marker, that is DNA barcode, they should have a low variation within the species and high variation between the species. What is this within the species, between the species? Here is a hierarchy, right? We have there are multiple species under same genus, multiple genus under same family, multiple families under order, class, phyla, kingdom. You know the hierarchy, taxonomic hierarchy. When I say low variation within species, when I compare 2 DNA sequences of Orisa sativa, so it is, say for example, it is 1% similar. When I compare the DNA sequence of Orisa sativa with Orisa bharti, it should be at least 3%. It is 1% variation is there. 99% similarity will be there within species. When I compare the sequences between sativa and bharti, it is more than 3% variation. That is maybe 96% similarity or 95% similarity. Okay, so intra-species variation should be relatively low when compared to the interspecies variation. This is the main characteristics for any gene sequences to act as a barcode. Okay, so what is this variations? Consider I have a Orisa sativa sequence here and another Orisa sativa sequence here. Okay, when I say for example, I have taken 100 base pair length. Okay, the base pair is a unit for measuring the DNA length, right? So 100 base pair length I have taken. When I compare 200 base pairs, if I have 1 base pair variation, it is 1% variation. 1 nucleotide in 100 base pair is variable, so it is 1 nucleotide variation. Okay, when I have no base pair variation, it is 0% variation or 100% similar. When you say 1% variation, it is 99% similar. When you say 100% similar, there is 0% variation. All right. So within the species, when you analyze DNA sequences of the same species, their variation should be low. When you analyze the DNA sequences of different species of same genera, their variation should be relatively high. So this is called species cutoff. When I say yeah, species cutoff of DNA barcode is 3%, what it means is the variations that is permitted within the same species is 3%, meaning they during blast analysis they should share more than 97% similarity. If any DNA sequence that share more than 97% similarity, I can call them as a same species. When they share less than 97, meaning 95% similarity, say I sequenced an Orisa leaf, I don't know what species it is. When I go to a blast search, when the sequence similarity is 95%, I am very sure it is Orisa, but it is not Sativa. When it say 95% similarity with Orisa sativa, it could be Orisa and not sativa. It can be Bharti or any other species under the genera Orisa. Okay. So likewise, we have species cutoff 3%, we have genus cutoff 5%, family cutoff, order cutoff, class cutoff. Likewise, in every hierarchical order, there is a boundary for this. Okay, so let's see these barcode genes. What are the features of the barcode genes? Like I told you, any gene to be act as a barcode, they should have a low intra-species variation and relatively high interspecies variation. In animal kingdom, entire animal biodiversity can be decoded with this one gene called cytochrome C oxidase gene, subunit 1 gene. We have subunit 2, 3. This is a mitochondrial genome. They are present in mitochondria. Since they are present in mitochondria, they are of multiple copies in number. You have a multiple mitochondria, and in every mitochondria, there will be multiple mitochondrial genome. So during PCR, since the number of copies of the barcode is high, you can easily amplify them. All right. And universal primers are available for that. Meaning, I don't care whether it's a human mitochondria or fish mitochondria. If I use the primer, it will work. It will bring out the cytochrome C oxidase subunit 1 gene despite whichever phyla we are going for sequencing. Okay, and they are of short sequence length. This attribute is very important, short sequence length, because this technology, like I said, has been proposed a decade back, during which most of our sequences ability is to have a continuous length of 900 base pair. I know now we have Novasec and other high-end sequencing platforms, but right back that time we have this 900 base pair or 800 base pair sequencing reaction. So it was very important to choose a barcode whose length is less than 800 or 700 base pair. So this cytochrome C oxidase is 650 base pair approximately in length, and they are because of these various consortia, now we have developed a huge data of cytochrome C oxidase. So anyone who is even a non-taxonomist in animals, they can simply have a tissue sample and get it barcoded and identify the species. So for plant barcoding, like I said, we have a chloroplast genome or MATK, maturus K gene. This maturus K plays a vital role in splicing, where they remove the introns post-transcription, and they are very much vital in expression of many genes associated with the photosynthesis. And we have this ribulose biscarboxylase L genes. Both individual genes or both in combination should be decoded, should be sequenced for plant species identification. See, we call them animal barcode as a cytochrome 1 gene, and the plant barcode as maturus K gene, RBCL gene. But for fungal barcode, we will call them as a ITS region. We will not call them as a gene because they are the combination of these 5 genes. ITS region starts from the partial region of 18S small subunit. Then we have a internal transcribed spacer 1, 5.8S gene, internal transcribed spacer 2, then 28S large subunit. So this 18S and 28S actually that's where the forward and reverse primer binds. They have a primer binding site. This region altogether called as an ITS region. Okay, and it is working fantastically for fungi. For prokaryotes, both for bacteria and archaea, we have the 6NS rRNA gene. We've been using this for more than 45 years now. Okay. So before I get into other applications, I just want to mention what is this barcode gap analysis. Okay, Barcode gap is something, I proposed in a general terms that 3% is a cutoff for species identification. Okay, this is not so in every species. Okay, maybe species cutoff for banana group of plants may be different than other forest plants. I don't know. Okay, every plant may have their own species cutoff, meaning you need to collect various varieties among single species to define this barcode gap. After analyzing and comparing each and every DNA sequence, you need to propose what is the average variations found among the sequences to call them as a barcode gap. Those who are looking for research article and easy, research topics to publish, defining barcode gaps for endemic plants of India can be an ideal topic to work on, because not much work has been done, and for many more group of plants this gap has to be defined. What is so big deal about having a species gap? Why should we have that? Okay, only when you have a species gap, only when you know what is the species gap of, say for example, Orisa Bharti, you can discover a new species. Yes, if you want to discover a new species, see, I know the barcode gap of, say, for example, Orisa Bharti is around 3%. When I think it's an Orisa Bharti, I go for sequencing. When I compare my sequences with the database, it shows 93% similarity with Orisa Bharti, meaning the maximum percentage of identity of all the sequences in the database is 93% similarity, meaning 7% variation is there. Now I know the sample I have is a new plant species. That's how the new species are. If not in zoology or botany, that's how it is done in microbiology. Okay, because in microbiology, for every species declaration, you need to produce a molecular data. But in case of plant biology, I feel there are many more plant species are yet to be barcoded. There are species we know for for many decades, but their barcodes are not available. In that case, when you barcode it, you can do a easy publication as first time barcode of that plant species. How do you know the plant species is first time barcoded? When you do the blast analysis, the percentage of variation will be more than 5 or 7%. That's why it's very important to define a barcode gap for every species, genera, family, class, vice versa. Okay. So what are the applications of DNA barcoding? Once you built a barcode library enhanced with local species population, now you can, even non-taxonomist anywhere anytime, they need not have any specialization. Now they can identify the species to some specimen to species level. We can now, because we are using a single gene to analyze the entire phyla or entire domain, now we can compare the phylogenetics between the organisms. We can do genetic distance analysis. We can do food authentication. When you buy a sardine cans, salted fish or preserved fish, what's the guarantee that every piece preserved belongs to same species or the species that is being declared in the can? We do not know. Now we know because of barcoding. Simply take a single fragment of a fish, go for sequencing, you will know what fish is that. Okay. Illegal trade monitoring. Even after fishing holiday or restricting the capture of red listed species, still many fishes are illegally exploited. The one easy tool fishermen had 20 years back is, once, say for example, dolphins are not for consumption, but when they are accidentally caught in the net, the fishermen are not ready to throw it away, because while pulling it in, they thought they had a gold mine caught. But when they discovered it's a dolphin, they will just go for cutting them into various pieces, skinning them out, preserve the flesh in the ice, bring it to the shore, thinking the monitoring authority cannot find it. But now we know, using barcoding, if I have a single 10 milligram of tissue is sufficient. 10 milligram. Just extract DNA PCR. I will go for CO1 sequencing. With that sequence, when I compare with the database, I know what fish they have caught. So likewise, this barcoding has opened us many avenues, many new applications. Initially, detecting an invasive species or identifying a cryptic species is very much challenging. With molecular data now, they are finding very easy. Their identification is facilitated very easily. Okay. So like I said, because of this barcoding database, now adulteration can be monitored, disease can be screened, These sequences can be used in biomonitoring and conservation services, dietary assessment. Okay, I will tell you, once you have a barcoding database enriched with local plant species sequences, now with simply collecting the fecal pellets of bat, I know what are the plants the bat is feeding. It can be very well decoded now because we have a database. We can simply dissolve the fecal pellet and go for entire DNA extraction, sequence it. We can decode what are the plant species the bat has consumed. Okay. And another fascinating application I find, I realized this on by myself through this publication. Conventionally, different species have different morphology. Same species will have similar morphology. It's a conventional fact, right? But see here, we were working with the deep sea polychaetes, marine worm, and when we went for sequencing, I was a molecular biologist in the team, and I was working with a nematode taxonomist, polychaete taxonomist. The taxonomists who've been working with polychaetes for more than 10 years confidently identified this specimen D and G as a different species. It is very much evident even for non-taxonomists. See the structure of this specimen. Then they have a cilia around their body. Cilia is some protruding appendages that is used to capture the oxygen in the water. Okay, and see the cilial structure of specimen D, which is very much extended from the body. So naturally, everyone thinks it's the same different species. But when I did the barcoding, they shared 100% sequence similarity, specimen D and G. Then we come to know we sample this from two different regions. When we sample this, the dissolved oxygen in the water is more than 3 milligram per liter. Here, the dissolved oxygen is less than 0.5 milligram per liter, meaning they are living in the oxygen deficient zone or oxygen minimum zone. So they have morphologically adapted to evolve the cilia and increase the surface area of the cilia to capture more oxygen from the water. You see such a morphological adaptation which can easily mislead the taxonomists to recognize them as a two different species can be resolved using DNA barcoding because they shared 100% similarity. Right. So enough about DNA barcoding. Now I want to introduce to the next level of barcoding. This is called meta-barcoding. Because we have generated the database enriched, say, for with plant sequences, now you don't even need to have an individual specimen collected for identification. You can simply collect a group of specimens, extract the community DNA, and go for PCR. In PCR, you amplify only the plant's MADK and Maturus RBCL gene sequences. Now you generate the sequences, compare with the database, you will know what is the individual species contained in that community sample. Okay, this is called meta-barcoding. Even the bat fecal analysis, what I told, is actually a meta-barcoding. You are taking the fecal pellet of the bat, extracting the entire DNA, even bat's DNA, microbial DNA, everything will be extracted. Only in the PCR, because of the specific barcode primers, we will amplify only RBCL genes and MADK genes, which are predominantly present in plant system. So only plant amplicon you will get. When you compare with the database, you will know what are the plant species that bat has been have consumed. So what is the main difference between barcoding and meta barcoding? This is the difference. In DNA barcoding, you will collect individual samples, extract DNA, sequence it, compare it with the database, you find the species. In meta barcoding, you can take the community samples, grind it well, extract the community DNA, and go for sequencing, compare the sequences. You have a next generation platform or cloning and sequencing to separate individual sequences, compare it with the database, you will find the species composition. It's not an idea, it's already happening. I have seen hundreds of papers coming in botany with the meta-barcoding concept, where the complex composition of processed food, plant foods, actually are subjected to meta-barcoding. Even ISO and FSSAI these days are demanding meta-barcoding data for authentication. All right. So DNA barcoding, meta barcoding. Now I want to realize the next stage of barcoding, that is eDNA barcoding, environmental DNA barcoding. This is the ultimate technology revolution, I would say, because now if you want to identify a plant sample, you don't even want to collect the plant sample. Yes, we have realized this. If I want to identify a seaweed composition of estuary where there are more seaweeds, just 100 ml of seawater is enough to find the seaweed composition. I can simply collect the seawater. I can filter it through a DNA binding membrane. All biological system, including seaweeds and fishes, when they grow, they excrete, they shed epithelial cells or old cells into the water, which will contain the DNA. So the entire ecosystem in aquatic, air or land can be sampled with soil, air or water. Their DNA can be easily sampled. That's the idea behind the eDNA barcoding, where they use a filter paper, DNA binding filter paper, to and pass on the water sample. That filter paper will be subjected to DNA isolation. From that filter paper, the PCR is conducted. Only RBCL and MATK genes are amplified here. They are subjected to data, they are compared with the database, and we will know what species are there in that ecosystem. There are many studies that have proven that fish DNA can be stable in seawater for 48 hours. Once a fish excrete, even the intestinal epithelial cells along with the excreta found or excreted out, that fish DNA can be stable in water, resistant to photolyase and many more parameter microbial degradation for up to 48 hours. Meaning, when you sample a seawater, you are actually seeing the activity happening in the seawater in around 48 hours. So the entire eDNA barcoding workflow goes something like this. You collect the sample, soil sample or air sample, just use a filter paper to filter the particles in the air, or the water sample to collect the environmental DNA. That's why it is called eDNA barcoding. And the second step is DNA extraction, targeted DNA amplification. See, another beautiful thing this eDNA barcoding did is, after collecting 1 liter of seawater, I can decode seaweed biodiversity. Now, I have a curiosity. What are the animal or vertebrate and invertebrate systems living in that ecosystem? Now, using the same DNA in the PCR, I will go for CO1 amplification. Now I can decode the entire CO1 diversity. If I use the ITS primer here in the PCR, I can decode the entire fungal diversity. If I use the 6NS RNA primer, I can decode the entire bacterial diversity. Just with 1 liter of seawater, entire ecosystem can be decoded. Starting from even viruses, if you want, you can decode it. Not collecting the actual specimen. This is called invasive sampling. Without collecting the actual specimen, the entire biodiversity can be decoded using bioinformatics and other comparative analysis databases. Okay. So is this something new? I'm proposing no. You see, the environmental journals and environmental sustainable journals, you will see they are actually flowing with the eDNA barcoding works. Here is an example. Airborne environmental DNA metabarcoding detects more diversity with less sampling effort than traditional community survey. This is the result they have found. They have chosen an area where traditional taxonomists went around to identify the trees, forbs, and grasses in the area. They have installed something like this, which can trap the air particles. Okay, they are air filters actually. The natural air that has been flowing around is getting trapped in the filters here. From this filter, they have extracted the DNA, went for targeted PCR, RBCL and MATK. They went for comparing with the database, and they found decoded the species. Here, see, eDNA were able to identify more than 13 species that is missed by traditional efforts. Okay, even for trees, they identified 4 species. 2 are very common with the traditional, and the traditional has missed 2 more species here. Okay, they are performing better than traditional methods. And this is one of our work where we try to use eDNA barcoding for identifying seaweeds. So what we did is we collected samples from southeast coast of India, Tuticorin to south, relatively northern part till Chennai. So the entire southeast coast was covered here. We filtered around 10 liters of seawater in the filter paper. From there, we collected the DNA and went for barcoding. We discovered around 31 species. Okay, when we compare it with the literature, not even 20 species are documented. 20 different species of seaweeds are known around the southeast coast. Never 31 species. Okay, so these are the sampling area. So we went for further analysis. I used RBCL gene for this barcoding effect. See, seaweeds are basically 3 groups. Brown seaweed, green seaweed, and red seaweed. precisely RBCL has clustered them in 3 different groups in the phylogram. This is something called phylogram. All phylograms should be read from the left hand side to right hand side. So by reading from left hand to right hand side, you know that there are 3 groups here. Group number 1, group number 2, branch number 1, branch number 2, and branch number 3. You see, not even single sequence was misplaced. The sequences denoted in orange is actually sequences generated in this study. The sequences in the black is actually a reference sequences taken from the NCBI database. All brown seaweeds are clustered in one branch, all green seaweeds are clustered in one branch, and all red seaweeds in one branch, meaning the intraspecies variation of RBCL of seaweeds are less when compared to interspecies, relatively less when compared to intergenera, interfamily, interclass. Only when the gene have such a characteristics, this kind of clustering is possible, right? So that's what happened, and this is the BLAST result I have given here. BLAST result is nothing but basic local alignment search tool, meaning you are searching your query sequence with the reference sequence by aligning them with the reference sequence. Okay, so see the first result. It is only 88%, 88.35% identical to sargassum, meaning it definitely a first seaweed that is not yet barcoded. Okay, and if you go to only 90% identical, and here 94% identical, so we know that using eDNA barcoding, we end up realizing that the barcode effort is very limited in this seaweeds. So what happened? We again went for actual DNA barcoding instead of eDNA. We again went to all these stations. We collected seaweeds with the appropriate permission, and we have generated the database now. And actually, the manuscript is being prepared, and it will be available very soon. Okay. eDNA barcoding is not new, like I said. Even in the herbal formulation preparations, they are using eDNA barcoding for authentication. See, they are simply collecting when the herbal formulations are prepared. The vapors that is carrying their DNA, they are actually collected through the DNA binding column, where the DNA molecules are collected during the herbal formulations. These DNA binding filter papers are actually used for DNA extraction, sequencing, and to authenticate that this is the herb they have advertised in the packet. Okay, like I said, FSSAI and ISO, they have mandated this now. Every batch of preparation should have a DNA binding column used and stored, which can be cross-verified later. Okay, this is very well happening in the industry, and these are the plant species they have identified from the DNA binding column. Okay, so anyway, this presentation will be available. If you want to have a longer look, feel free to check the video again. So this is what I told. The main advantage of eDNA barcoding is if you have a 1 liter of sample or 1 gram of soil, you can decode from microbes to macrobes. Whatever have lived in that ecosystem can be decoded with just collecting the environmental sample. Okay, so here is a global effort where they took the sea sample around the globe. 1 liter seawater was used in every sampling point. This picture shows the sampling point done around the globe. The yellow color is actually a total biodiversity mapped. Domain specific is shown in the green color. Kingdom specific is blue color. Phylum specific is the black dots, and targeted assessments are shown. You will agree with me, based on the size, you see, when we generate more eDNA barcodes, actually we are able to identify them only to the domain or kingdom specific, which is very much evident from this picture. Those black dots size are relatively smaller to compared with the blue or green dots, right? So this is why, if you want to explore this technology in full-fledged form, we need to initiate more barcoding efforts, especially the local species what we have. We need to barcode them so that they can find application in various industries, and they may evolve into something even more beautiful. Okay, so these are the I just want to show the sneak peek of what's happening in the DNA barcoding and its application related to plant systems. They are using for dietary supplement confirmations, authentications, even commercial products. These days they need to produce the molecular data for getting authenticated from the authorities. Even the herbal market, if you want to authenticate or revalidate a particular herbal formulation, you can use this DNA barcoding. All these communications are related to that. Okay, let's talk little business about DNA barcoding. It is fastly growing industry, I would say. It's a DNA barcoding technology. You can advertise like this because it's a $12.5 billion industry that is growing 16.5% annually. Even one survey has predicted to grow $40.5 billion by 2032. Okay, so it has a very potential market now, right now, even in near future. In India, even it is much more faster. The market is around $1,410 million, and they are increasing by 26.7% annually. So I would say UG and PG students who are looking for job can think about this. Okay, since they have a huge market potential, now you can orient yourself what to be skilled with, what kind of project to do, what kind of thesis to produce, so that it can resonate with what's happening in the market, and you may find a job or research with ease. Okay. So I just want to introduce this company because they are doing a very good job in DNA barcoding. Okay, their intention is to increase the barcode lab size. Barcode database. They are very much into decoding the local diversity. In my estimate, they may have produced more than 10,000 barcodes in last 15 years time. Okay, they are founded in 2013, and they are doing very good, excellent services. This is their infrastructure. They have a megabase sequencer that can handle 100 reactions per 5 hours, and they have a ABI sequencer 3700XL which can handle 1,000 sequences per day. And they have a primer synthesizing machine, Mermaid machine. This is a bread supplier for the company. I know them. And here is a PCR workstations where even 770 samples can run in. So, as I told, they are into promoting DNA barcoding. I can vouch for them. They are the most affordable DNA barcoding services in the country. They do herbal formulation authentication. They do microbial identification, mushroom identification. They also do metabarcoding services. Okay, and if you are going for multi-locus gene sequencing, they can get you in 3 days time. Like I said, we need to use both MATK and RBCL for precise species identification in many cases. They do forage identification, insect species identification, and they have this educational channel too. Since it is available in your YouTube, you can check it out. They are publishing videos how they are actually processing the sample, how the sequences are generated, how to handle the machine output files, how the machine output files are subjected to blast analysis, how they are using DNA sequence to find the species name. You can see, and you can follow them to learn more. Okay, I see this is the current status of barcoding efforts in India. By 2021, this paper was published. The bubble size represents the quantity of barcodes that are being generated worldwide. See Indian scenario. Okay, it is very much in primitive stage. I should say that I most of my publications are in DNA barcoding, and I'm not any taxonomist. I'm a microbiologist. My entire interest lie with studying microorganisms, but most of my publications are in DNA barcoding. I will tell you how it is happening. Okay, once you have a particular species sequenced, when you find during blast analysis that your sequences share has only 93% similarity or 94% similarity, meaning you have barcoded it for very first time. Because these efforts are very much less in India, you can write a paper something like this: first time barcode of that plant species. The journals will accept you immediately just with one DNA sequence data. This can be done because it's a new data. Journal's objective is to promote new data. They will go for it. This is very decent journal. Okay, around 4 impact, I guess. Still, they got this one. There's one sequences has been published. Another irony is, the world or the countries with high biodiversity actually are producing low barcoding data, and countries with low biodiversity, relatively low biodiversity, they are very good in cutting edge technology. Okay, we need to bridge this gap. Okay, so I would say I thought since I'm delivering this talk to Department of Plant Biology and Plant Biotechnology, I thought I will give a try and see whether if we have barcoded all the endemic species of India, endemic plant species. I don't know about this Sophora interrupta, but I just gave a try. Okay, I went to NCBI, I selected the nucleotide, I gave this name Sophora interrupta, this plant species. No items found, meaning they have not been barcoded. Not only barcode genes, none of the gene in this species has been barcoded, meaning if you can barcode this species, one sequence is sufficient enough to publish it in a journal. First time barcode of Sophora interrupta, that's it. You can push it to the journal. So I thought I will spend some time to see whether all endemic species of India are barcoded or not. All, at least 20, top 20 species are barcoded or not. The reason I go with endemic species is I thought since it is endemic, the chances of other country people, the countries with high-end technology to access this sample will be less. So I thought I will generate this list of 20 species. I will check it in NCBI. To my surprise, I just spent 20 minutes time. This data is as of July 4th, 2 a.m. Okay, 8 species that is being highlighted in the red. I don't know about this plant species, but they are not yet barcoded. That's what NCBI search tells me. These pictures corresponds to the red these species that are yet to be barcoded. Meaning, if you just barcode this species, you very well do a simple publication. Okay, see here. This is one of my paper. I'm not a isopod taxonomist, but this paper is in giant isopod. Dr. Shankar here is actually a taxonomist of an isopod. I was working with him. I was doing his collection, DNA isolation, PCR, sequencing, analyzing the sequence, preparing the manuscript. Where I noticed that this Bathynomus species, 2 species, they have nearly 95% similarity, meaning they have not been barcoded before. So we simply wrote our paper, first time barcodes of Bathynomus 2 species. They just got clicked. I worked in this project for 25 days and I wrote a paper in 5 days, communicated, waited for 25 days, got published in regional studies. And I think it has around 2 impact or something. So when you actually enhance the local diversity, when you sequence the species yet to be barcoded or not yet barcoded, we may end up publishing them very easily. Okay. So I want to introduce this. This is something being offered by Barcode Biotechnology right now. They are conducting a summer internship. Like I said, this company works with difference. They are not into commercial aspect, even though they are charging a small fees for this internship. What they are actually doing is they are promoting the barcode of not yet barcoded species. So in this internship, they are requesting students to choose a sample, plant sample or any sample. If you have chosen a sample, student or even faculty members, postdocs can participate in this internship. It is an online internship. If you can choose one sample which is completely not yet barcoded by doing the search in NCBI and then choose it, which is easily accessible to you in the locality, you may end up getting free sequencing from this during this internship. And I guess using your own data, they will teach you the bioinformatics analysis, how to analyze and how to go for it. Okay. So since this company is sponsoring me in many ways, I thought I will pitch their this internship opportunity here. This is not something new they are doing. They do this every time. These are the various free sequences that is being published in NCBI. Yes, you will happen to publish your sequence in NCBI. You will also happen to write a manuscript and communicate it. They will give a full support in that. So I hope I started with what is barcoding, and I have the entire mechanism behind the barcoding has been decoded. We slowly moved into what is metabarcoding, and then we moved into eDNA barcoding. I don't want to stop there. Barcoding is something even you can do. It is very much accessible, and it is there. This company is operating in Chennai. So I know everyone can access this, and I want to introduce this opportunity so that everyone can do some barcoding. So thank you very much for having me today. Happy barcoding. If you guys have any questions or anything,
Thank you, sir. Thank you so much. And I think it will give more idea about to students, sir, because what I thought is you will give basic ideas only, but you can suggest many ideas to research work also. I think so. But now I would like to invite Dr. S. Sitaraman sir to put up hands.
Sir, good afternoon, Dr. Prasanna here. I would like to extend A heartfelt vote of thanks to each and every one of you making our barcoding program and a grand success. Your present support and contributions have been invaluable, sir. To our principal, Dr. N. Sujata ma'am, and management, we appreciate your unwavering support and encouragement in the organizing this program. Your visions are academic and excellence and innovations is truly inspiring. To our esteemed chief guest, Dr. Prasanna Kumar sir, thank you for your sharing experience and insights. Hope that knowledge and skill gained the benefits you and futures of endeavors. Thank you for 100%. Thank you.
Thank you, sir. Session
Thank you,
is
sir.
over.
Yes, yes, sir. Thank you.
Kindly join me. Wait a minute.
Postman.